Hi friends, I am Vasundhara from BZC second year. I am currently pursuing Bachelor of Science in Telangana Tribal Welfare Residential Degree College at Mulugu. My subject is Botany and my topic is structure of embryo sac. Today we are going to discuss about structure of embryo sac. What is embryo sac? This embryo sac belongs to the embryology. The study of development of embryo is called embryology. The father of embryology is the greatest scientist 𝗳𝗧𝗦𝗔𝗧. He was born in 1904. Now today we are going to discuss about contents of embryo sac. First we discuss about introduction of embryo sac, and next we will learn about learning objective, and next we study about structure of megaspores, and next we talk about structure of embryo sac, and next we will talk about components of embryo sac. In these components, how many components are present in embryo sac and what are the components of main functions? This all comes under into the components of embryo sac. And lastly, we discuss about types of embryo sac. Now coming to the topic, the embryo sac. Where it present embryo sac? Yes, embryo sac is present in flowering plants inside of the ovule. The other name of embryo sac is female gametophyte. The female gametophyte of angiosperm is called embryo sac. Here the angiosperm. Angiosperm derived two words. One is angio, another word is sperm. Angio means closed, sperm means seed. So it is called angiosperm. We know that ovule is covered with ovary. After fertilization of ovule gives seed, and after fertilization of ovary gives fruit. And embryo sac is the most common type of 7-celled and 8-nucleated structure. Why we are called 7-celled and 8-nucleated structure? Because as we seen in diagram of embryo sac, here upper part has and lower part has. In the upper part is upper part of micropylar end has 3 cells, and lower part of a chelagel end has 3 cells, and middle of the 1 cell is present. So 3 plus 1 plus 3, 6 cells are present. So 7 cells are present. So it is called 7-celled embryo sac. And next, why we are called 8-nucleated structure? Because as we seen in diagram of embryo sac, in upper part of a micropylar end has 3 cells, and lower part of chelagel end has 3 cells, and middle of the embryo sac has a 2 nucleus. 3 plus 2 plus 3, we get 8 nucleus. So it is called 8-nucleated structure. The embryo sac is a 7-celled and 8-nucleated structure. And most common type of embryo sac is a polygonum type. Why we are called polygonum type? Yes, it was first observed by Stanzberger in the plant of Polygonum species. So it is called polygonum type in the year of 1879. And female gametophyte is a primitive or normal type. And next we will learn about learning objectives. In this learning objective, we learn to get an idea about an embryo sac, to know the concept regarding the formation of embryo sac, and lastly we discuss about the different types of embryo sac. And next, structure of embryo sac and its components. Embryo sac is mainly containing 3 components. It is egg apparatus, central cell and antipodals. And last we discuss about other aspects of embryo sac. See here, it is the structure of megaspores. How form megaspore? Means 2 nucleus or MMC. MMC means megaspore mother cells undergoes metatic division, then form 4 tetrad cells. It is undergoes again further metatic division, then form 8 nucleus. These 8 nucleus are 3 cells are arranged in micropylar end, and 3 cells are arranged in chelagel end, and remaining 2 cells are arranged in center of the embryo sac. Lastly we observed formation of a embryo sac. See here, I think all you know this diagram. Yes, it is the diagrammatically view of embryo sac. In the embryo sac, upper part is a micropylar end and lower part is a chelagel end. In upper part of micropylar end has a 3 cells, and lower part of chelagel end has a 3 cells. In the upper part of micropylar end has 3 cells. They are 1 egg apparatus, 2 synergies, and lower part of chelagel end has a 3 cells. These 3 cells are called antipodals, and middle of the embryo sac has a 2 nucleus. These nucleus are called polar nucleus. In the synergies appears presence of a finger-like projection or cellular thickness. It is known as a filiform apparatus. Filiform apparatus. Next, we will discuss about egg apparatus, first components of embryo sac. 3 nuclear group put together at micropylar end, it is known as egg apparatus. In egg apparatus, it contains one egg cell, 2 synergies. In the egg apparatus, egg cell is the biggest cell and synergies are a smallest cell. In the synergies, finger-like projection at micropylar end is called filiform apparatus. And function, what is the filiform apparatus function? Means the main function of filiform apparatus is it guide the pollen tube enter into synergies. Why it guide the pollen tube enter into synergies? Because in the pollen tube has a 2 male gamete. One male gamete enters into the embryo sac and fuse with secondary nucleus, then after undergoes fertilization. That's why it guide the pollen tube enter into the synergies. And next function is it observe and condensation of food material from new cellulose to embryo sac. Here the new cellulose. What is the new cellulose? New cellulose means it is the help for the transportation of food material to embryo sac. And new cellulose is a diploid in stage. In the egg apparatus, upper side has a nucleus and lower side has a vacuole. Is present in synergies. And in the same case, in the egg cell we observed in upper part has a vacuole and lower part has a nucleus. And next, see here, it is the formation of a egg cell. Formation of a egg cell. In this formation of a egg cells, structure of ovule is present. In this ovule have a nucellus, integments and MMC. In this nucellus, after post fertilization, they undergoes completely or absorb the food material. And integments are undergoes after fertilization, they are formed seed coat. And MMC, megaspore mother cells, then undergoes meiosis division, then form 4 haploid cells. Among these 4 haploid cells, 3 cells are degenerated and only 1 cell is a functional cell. And this 1 cell is functional cells undergoes 3 times free nuclear metatic division, then form 8 haploid nucleus. In this 8 haploid nucleus, 2 cells are polar nucleus and 1 cell is egg cells and another remaining 3 cells are antipodals. And lastly we observe male cells of the main cells of the polar nucleus 2 and 1 is the egg cell. And next we discuss about the central cell, second component of embryo sac, that is central cell. The central cell is the biggest cell of the embryo sac and it has the 2 polar nucleus fused to form secondary nucleus. This secondary nucleus are undergoes 2n division. 2n division means here the central cell is formed, one is the micropylar end and one is the 𝗧-𝗦𝗔𝗧 end. These 2 cells are called polar nucleus. This fused to form it is called secondary nucleus. The fusion of polar nucleus is before or after entry of the pollen into embryo sac. The central cell is found be connected with egg, synergies, antipodals, plasma-desmata. Central cell is rich with all cell organelle like mitochondria, chloroplast, and central cell is the active metabolism. Next, we will observe the central cell in diagram. Central cell is present in the middle of the embryo sac. Here the polar nucleus, it is present in middle of the embryo sac. One cell is come from the micropylar end and another cell is come from the 𝗧-𝗦𝗔𝗧 end. These 2 cells are called polar nucleus and it fused to form 2n secondary nucleus because 2n means one cell is haploid and another cell is haploid to form diploid stage. So it is called 2n secondary nucleus. Next we observe last components of embryo sac, that is antipodals. 3 nuclear group put together at 𝗧-𝗦𝗔𝗧 end, it is called antipodals. In the antipodals, some plants have a many antipodals. We taken as the best example grasses. 3 antipodal cells with variable nuclear condition, and antipodals are help for nourishment of embryo sac. And here observe other aspects of embryo sac. Haustoria. What is the haustoria? Haustoria means in the embryo sac, some cases it shows growth behind the ovular tissue, or either micropylar extensions or 𝗧𝗦𝗔𝗧 extension. These two extension is called haustorium. Example, Santillaceae, Oriantheaceae, Acanthaceae. Perisperm. What is perisperm? Yes, perisperm means mass of a nutritive material outside of the embryo sac. It is called perisperm. For example, black pepper, maize, groundnut nut. And the embryo sac is structurally, functionally well organized to nurture the growing embryo. See here, it is the present the middle of the perisperm. This perisperm is the new cells remaining the thin layer is called perisperm. It is covered with the seed coat. Now coming to the last topic, types of embryo sac or female gametophyte. According to the Acharya Panchavan Maheshwari, the female gametophyte has a 7-celled and 8-nucleated structure. The number of megaspores taking part in the development of embryo sac. The number of divisions occurring in the nucleus of the functional megaspores. Organization of nucleus in the mature embryo sac. Mainly embryo sac has 3 types. They are one monosporic embryo sac, bisporic embryo sac. Now we see in the monosporic embryo sac and bisporic embryo sac. How to formation of monosporic embryo sac means, first we observed first cell MMC megaspore mother cells undergoes metatic division, then form 2 cells. These 2 cells are called dyad. In this dyad, one is functional and another is the degenerated. Then after one cell is undergoes metatic division, then form 2 nucleus. The 2 nucleuses undergo meiosis division, then form 4 nucleus. These 4 nucleus are undergoes metatic division, then form 8 nucleus. 8 nucleus are arranged in 3 nucleus are micropylar end and 3 nucleus are 𝗧𝗦𝗔𝗧 end. Remaining 2 cells are middle of the embryo sac. It is the formation of monosporic embryo sac. And next coming to the bisporic embryo sac. Bi means 2. So one is the functional megaspore undergoes metatic division and then form 2 cell. It is called dyad. If dyad undergoes metatic division, then form 4 cells. These 4 cells are undergo metatic division, then form 3 cells are degenerated and only 1 cell is the functional cells. If functional cell is undergoes metatic division, then form 2 cells. The 2 cells are undergoes metatic division, then form 4 cells. These 4 cells are undergoes further metatic division, then form 8 cells. 8 cells are arranged in the micropylar end 3 cells and 𝗧𝗦𝗔𝗧 end 3 cells. Remaining 2 cells are arranged in the polar nucleus, middle of the embryo sac. And last we discuss about the fritillaria type, tetraspore type. In this first MMC megaspore mother cells undergoes metatic division, then form 2 cells. The 2 cells are undergoes metatic division, then form, it is the arrangement of a 1+3 arrangement. So the micropylar end has a 1 cells and 𝗧𝗦𝗔𝗧 end has a 3 cells. This cell undergoes metatic division, then form micropylar has a 2 cells and 𝗧𝗦𝗔𝗧 end has a 6 cells. The 6 cells are undergoes metatic division, then form in the micropylar end has a 4 nucleus and 𝗧𝗦𝗔𝗧 end has a 12 nucleus. Then we observe lastly formation of a embryo sac, 7-celled and 8-nucleated structure. And coming to the monosporic embryo sac. Monosporic embryo sac has 2 types. One is the allium type and endomian type. Allium type means it is the first observed the Stansberger. It is the only the functional megaspore is formed 7-celled. It is called allium type. And next we observe the tetrasporic type. In the tetrasporic type has a 7 types. One is the Plambagella, Pinia and Fritillaria. These are the few of the tetrasporic type. And it is the main concept and important also exam. So well prepared. And we discuss about the types of embryo sac. In the types of embryo sac, in the monosporic embryo sac has a 2 types. One is the allium type and another one is the endomian type. In allium type has a It was which functional megaspores and diode cell, and endomian type has a it is the functional diode cell. In bispore it is the tetraspore. Lastly, I conclude that the embryo sac is 7-celled and 8-nucleated structure. 8-nucleated structure and an integrated approach to studying the female gametophyte of angiosperm and revolves the fresh information on the mechanism of sexual reproduction in higher plants. And to answer concerns generated by morphological investigations and research are using genetics, molecular and physiological techniques. These are the type of the embryo sac has a important question to the exams, and it is asked also in the bits. And we observe the embryo sac is a, we learn today session is introduction of embryo sac and learning objectives and structure of megaspore and structure of embryo sac, components of embryo sac and types of embryo sac. And embryo sac is a 7-celled and 8-nucleated structure because in the micropylar end has a 3 cells and 𝗧-𝗦𝗔𝗧 end has a 3 cells. 𝗧-𝗦𝗔𝗧 end has a 3 cells and center of the embryo sac has a 1 cells. Total 7-celled is formed, so it is called 7-celled and 8-nucleated structure because in micropylar end has a 3 cells and 𝗧-𝗦𝗔𝗧 end has a 3 cells and middle of the embryo sac has a 2 nucleus. So 8 nucleus are formed. It is called 8-nucleated structure. So embryo sac is a 7-celled and 8-nucleated structure. See here, it is the diagrammatically view of embryo sac. In the embryo sac, mainly three components are present. One is egg apparatus and central cell and antipodals. In egg apparatus, one egg cell is present and 2 synergies present. In egg cell, in synergies we observe the finger-like projection or filiform apparatus. It is called egg apparatus. In middle of the embryo sac has a polar nucleus. It is called the secondary nucleus. In this diagram we observe in the lower part of a embryo sac, it has a 𝗧-𝗦𝗔𝗧 end. It is known as the antipodals. In this diagram we see the red color, it is the polar nucleus. It is called polar nucleus. In this synergies, presence of a finger-like projection or cellular thickness, it is called the filiform apparatus. And see here structure of megaspore. How to form megaspore means, first 2 nucleus are undergoes metatic division, then form 4 cells. These 4 cells are undergoes metatic division, then form 8 nucleus. The 8 nucleus are undergoes metatic division are replaced by 3 cells are micropylar end and 3 cells are 𝗧-𝗦𝗔𝗧 end, and the remaining 2 cells are middle of the embryo sac. Lastly, we observe the embryo sac is formed. See here, it is the formation of a egg cell. How to formation of a egg cell means, here the structure of a ovule. In this ovule, new cells and integments are present. New cells are after post fertilizations, they gives to the totally or observed food material. Integments are after they formed the seed coat. And MMC, megaspore mother cells undergoes meiosis division and 4 cells are then form 4 haploid cells. Among the 4 haploid cells are 1 cells undergoes metatic division, then form 8 haploid nucleus. In this 8 haploid nucleus, 2 polar nucleus form and 1 egg cell is formed. Lastly, we observe main cells, that is 2 polar nucleus, 1 is egg cell. Lastly, I conclude that The embryo sac has 7-celled and 8-nucleated structure. An integrated approach to studying the female gametophyte of angiosperm. It involves fresh information on the mechanism of sexual reproduction in higher plants like angiosperm. And to answer concerns generated by morphological investigations, research are using genetics, molecular and physiological techniques. And thank you for this given golden opportunity to Secretary Sir and TSAT management.
ఆహారం పాడకుండా ఉంచడం ఆహార పరీక్షణ ద్వారానే సాధ్యం. మనం ప్రతిరోజు ఎన్నో ఆహార పదార్థాలు వాడుతుంటాం. అవి కొన్ని రోజులు